to talk a little bit about restriction enzymes, since those really are at the heart of most electrophoresis applications. So in the crime lab, in a paternity test, or even just in standard research when you're trying to verify the function or the presence of a gene, we're going to use restriction enzymes. And so I'll use the restriction enzyme called EcoR1. That's Roman numeral 1. And EcoR1 searches for this sequence, GAATTC. And DNA, as you know, is double-stranded and anti-parallel, A with T, C with G. And so when we read this top strand left to right, GAATTC, the bottom strand is going to read right to left because this is the front of the other strand. So GAATTC, this is called a palindrome, where the words or the segments read the same front to back. And so this restriction enzyme is going to cut this DNA like this. Okay? And we know that humans have about 99% the same DNA. You and me and your neighbor, we all have about 99.9% same DNA. However, that little 0.1% is going to vary. And so my sequence might read this, okay, with a few extra base pairs at the end. And yours might read that with lots of extra base pairs at the end. And so when this gets cut here, I'm going to have a very small fragment and then a very long fragment here. However, yours is going to have this same small fragment, but an even longer fragment than me. And so when these come out on gel, my two fragments are going to be a short fragment, or excuse me, a long fragment, and then a short fragment. But remember, your long fragment is going to be even longer, so it's going to be farther up the gel, and then that fragment will be the same. So the negative end and the positive end. And so again, we are human. We should have some matches. Okay? But this other difference is that small variability that the crime lab or the paternity testing uses to find those variable tandem repeat sequences and the restriction fragment length polymorphisms that are what separate each of us from in the human population one from the other. Remembering that identical twins should be the only people that would have the same DNA profile. All right, so I'm going to talk about some actual gel results from one of the electrophoresis that I ran, and this is the one that was the pre-purchase kit. And so when your students look at those de-stained gels, they're going to have 6 wells worth of data. Okay, these first 2 are the crime scene. The second 2 are suspect 1, and then these last 2 are suspect 2. And this is going to be enzyme 1, enzyme 2, enzyme 1, enzyme 2, and enzyme 1, enzyme 2. And so in the forensic world, they're going to use multiple enzymes, and you're actually going to see the evidence for why we do that after I draw the results here on the gel. In the forensic world, here in the United States, they use CODIS, the database that stores DNA information, and so they're going to use approximately somewhere between 12 and 17 different regions of the human genome to compare. And so that's the minimum number for it to be valid in the court of law. And so I'm going to show you why we need to use at least 2 enzymes to do some comparisons. And so in this lab, you're actually going to see 2 bands for each one. So here's our crime scene. Okay, and you can see here's one enzyme and with another enzyme. So this is the exact same DNA sample, but cut with 2 different enzymes. And so you get 2 different banding patterns from the same individual. Okay, suspect 1's DNA with enzyme 1, okay, matches the crime scene. Cut with enzyme 2, okay, and then, which also matches the crime scene, but for enzyme 1. Suspect 2, enzyme 1, also matches the crime scene. If we stopped there and only used 1 enzyme, we would not be able to differentiate which suspect had left behind their DNA sample at the crime scene. So in order to help us differentiate this, by using the second enzyme, we in fact see that it was suspect 2 whose DNA was found at the scene of the crime. And so the forensic investigator may have to testify in court or provide a report to the judge about their results here. Again, making sure to reference all the databases used to pull the DNA samples and all the comparative software and the controls used there in the lab. And so again, restriction enzymes are going to cut the RFLPs, or restriction fragment length polymorphism regions of the human genome, of which there are many hundreds. However, CODIS, again, only uses about 12 to 17 of those regions, depending on the lab that you're working with.